So good morning to all. Now we will start technical session first, genetic resource conservation, utilization and enhancement. In this technical session, Chairman Dr. A.K. Singh ji, Director IRI New Delhi, Co-Chairman Professor EVD Sastry sir, HOD PBG, College of Agriculture, CIE Imphal, then Rapporteur Dr. P.N. Shivlingam and Dr. Madhu Patial. In this session, first keynote address by Dr. A.K. Singh ji, Director IRI New Delhi. So regarding biodata, I read the first biodata. Dr. A.K. Singh ji. Dr. A.K. Singh ji, born in farmer family in village Bharat, Gazipur, UP. He did his graduation and post-graduation in genetic and plant breeding from Banaras Hindu University and PhD in genetics from IRI New Delhi. Dr. Singh joined as a director ICR IRI New Delhi on 18 January 2020. Earlier, he worked as a joint director, research in charge and head division of genetics, ICR IRI New Delhi. Dr. Singh has been actively involved in Basmati rice improvement for the last 2 decades and half decade. He is also associated with these improvement Basmati variety occupied about 1.8 million area, bringing prosperity to millions of Basmati farmers while earning 2,005 crores of foreign exchange annually. He has been associated with teaching of 3 postgraduate courses in genetics for last 20 years, guiding 15 PhD, 5 MSc students, with 3 of them winning the prestigious ICR Jawaharlal Nehru Award, 7 of them awarded with IRI merit medals. Dr. Singh has published 120 peer-reviewed research publications in rice genetics, molecular breeding, and grain quality. He co-authored a book entitled Marker Assistant Plant Breeding Principles and Practices, published by Springer. Dr. Singh pioneered the public-private partnership model for commercialization, first super fine grain aromatic rice hybrid, USA RS10, and several Basmati rice variety generating revenue of Rs 2 crore. He has served as an expert member in DBT, APEDA, RSC of NRRI and other institutes. He has been awarded with prestigious ICR Rafi Ahmed Kidwi Award, ICR Bharat Ratna, Dr. C. Subramaniam Award during 2013. He has also been recognized with IRI Best Teacher Award and other also many awards received. Not taking much time, I request to Dr. A.K. Singh ji to start your presentation and share your PowerPoint.
सर नमस्कार।
हाँ हाँ बोलिए ना
सर नमस्कार।
प्रेजेंटेशन करो।
Thank you, Dr. Haldhar, for your generous introduction. At the outset, I would like to thank the Honorable Vice Chancellor, Dr. Premjit Singh, Dr. Indira, Dr. Hazarika, Dr. Shastri, for inviting me to this program for giving the keynote address. I'm also thankful to the organizers for giving me this opportunity. We have Dr. Pawar, Dr. Balraj Singh, Dr. Bilal, all very distinguished people who have joined on this conference, and we heard our Honorable Minister who so elaborately described about the various schemes of government and how we can make use of those schemes, particularly in the northeastern region of the country, to enhance farmers' income. Is something that is we are going to discuss next 3 days. My presentation is not really very relevant to northeastern region, but the technology that we have used in improving the Basmati rice varieties and the technologies of using molecular breeding for transferring genes for assistance to biotic, abiotic stresses, for enhancing Basmati rice yield, and quality, and also resistance to biotic abiotic stresses. Is the experience that I am going to share with me. One of my PhD student has recently, she is about to submit her thesis, Bhuvaneshwari. She works at the same place in your university campus, in the ICR Institute. And Bhuvaneshwari has worked on a very relevant problem of making Shakao Paritan, the black rice, which is a GI protected commodity from northeastern region, particularly from Manipur black rice. She has been able to make it dwarf. And by making it dwarf, it is possible to improve the productivity maybe 2-3 times as compared to traditional Shakao Paritan. So that is something very, very important. And I will start by this presentation, how we have used molecular breeding in case of basmati rice for its genetic improvement. If you look at the traditional varieties of basmati rice, they are very tall, about 160 centimeter in height. They are prone to lodging. They are photoperiod sensitive, and their yield was hardly 2.5 tons per hectare. The Basmati rice breeding program in IRI was started in late 60s with the vision of Dr. Swaminathan to combine the quality of traditional Basmati into high-yielding background. And exactly the same model can be replicated for improving the productivity of Shakao Paritan, which is very much indigenous to Manipur state. So the first dwarf variety of Basmati rice was released in 1989 in the name Pusa Basmati 1. And in 2003, we released Pusa Basmati 1121. And this variety is currently grown in about 0.9 million hectare area. And in 2007, we came out with improved Pusa Basmati resistance to bacterial blight. And 2013, a variety Pusa Basmati 1509 has been released, which has short duration of 125 days, very high productivity, and it saves about 30% water, and at the same time, it provides opportunity for multiple cropping. The farmers in Punjab, they grow 1509 followed by potato, and then followed by chili. And that kind of cropping system, high intensity agriculture, brings about 4 to 5 lakhs rupees per hectare net income to these farmers. If you see over a period of time how this improvement has taken place, in 1930, the first Basmati variety, Basmati 370, was released, and its grain length on cooking is about 12 to 30 millimeter. But if you look at 1509, which has got a grain length of almost 1 inch, 25 millimeter on cooking. So this gives very high volume expansion, and this was the winning point of 1121, which was developed by the predecessor, Dr. V.P. Singh. We were associated in the team. And now 1509 has replaced 1121 in almost 50% area. The overall impact at the duration of traditional Basmati was reduced from 160 days to 120 days in the dwarf varieties, and productivity was improved from 2 tons per hectare to 6 tons per hectare. Now, generally in a plant breeding program, you don't find this seizure-like curve. You will have increase in yield associated with higher productivity, but here we were able to reduce duration and increase yield at the same time. So it was a negative association that was broken. And a recent variety that we have developed, 1718, which is improved version of 1121 with bacterial blight resistance, is picking up very well, and the total Basmati rice export earning annually is about 32,000 crores from these varieties. The major challenges which we have addressed through molecular breeding are the several biotic stresses: bacterial blight caused by Xanthomonas campestris, Pathovarroraceae, blast caused by Manipur tigricia. Then brown plant hopper is a tiny insect which sucks the sap from plants, and then after that the whole field will dry and you will have nothing to harvest. Teeth blight is another major problem which farmers will have to use fungicides number of times. And in addition to these problems, we have the problem of stem borer, we have problem of Bacane disease, we have problem of brown spot, false smut. These are the major diseases, and because of that, rice is one of the biggest consumer of insecticide, pesticide. This has resulted into one issue, particularly European Union, which is a major market for basmati rice export from India. About 6,000 crores rupees goes to European Union. But EU has become very stringent in terms of their pesticide residue limits. The MRL, maximum residue limit for tricyclazole, a fungicide which is used for controlling blast disease in rice, is brought down to 0.01 ppm. So it is not possible, as a matter of fact, if you are using fungicides. So the only way we can meet these targets to sustain our export, because one side, when our Honorable Prime Minister is focusing on doubling the agri export from 39 billion US dollar to 80 billion US dollar, these kinds of trade barriers which are imposed by many countries would become a deterring factors, and therefore we have to fortify our variety with genetic resistance so that you don't need to spray fungicide, and if you don't need to spray fungicides, the question of residue will not come. So we have followed a standard procedure of marker-assisted backcross breeding, and this will hold good for any crop, for any place, whether a student has to do it in Manipur or in Delhi, the procedure remains the same. The marker-assisted backcross breeding, you all know about the backcross breeding. Those students who are from BSc agriculture background and genetics students, you will particularly see that you have a recurrent parent, which is a very good variety otherwise, but it lacks into one or two characters. And you have a donor parent which has that character which is missing in recurrent parent is present in donor parent, but it has all other characters are not so good. So we make a cross and then we go for back crossing. Generally, the back cross breeding used to take about 6 to 7 years, very long time. But through molecular marker assisted back cross breeding, which consists of these 4 steps. The first one is foreground selection, which means the selection for the target locus, the gene that you want to introgress. Recombinant selection is elimination of linkage back. Whenever you bring a gene from outside, from a donor parent, then along with the genes associated flanking genomic regions also come. So you have to minimize that genomic region, and that is called recombinant selection. And background selection is about recovery of recurrent parent genome. And then phenotypic selection for the trait in which you are interested. So if you follow this integrated approach of molecular marker assisted backcross breeding, then you can expedite the backcross breeding program from just in 2 to 3 years. And if each year, if you take 2 crop season, you can achieve in 2 to 3 years the conversion of a otherwise good variety with the specific defect it has can be rectified. So we followed this approach, and the first variety we released was in 2007 in the name Improved Pusa Basmati 1. Pusa Basmati 1 was our earlier variety. It was susceptible to bacterial blight. In this, 2 genes XA13, XA21 were incorporated, and this variety was released for commercial cultivation. Subsequently, Pusa Basmati 1121, which is again a very good variety, but it also has susceptibility to bacterial blight disease. We transferred the gene from 1416 to 1121, and a new variety called Pusa Basmati 1780. It is a near-isogenic line of Pusa Basmati 1121, excellent grain and cooking quality. You can see the cooked rice picture, almost 1 inch in length. And this 1718 has been released for commercial cultivation, and this is replacing past Pusa Basmati 1121. It has got very sturdy stem, higher yield of 4 to 5 quintals per acre, more than that of Pusa Basmati 1121. Yet another variety, Pusa Basmati 6, that we had released earlier in the name Pusa 1401. This was also susceptible to bacterial blight, and we incorporated gene XA13, XA21 in this. And new variety, the field in which we are standing, is Pusa Basmati 1728, which has been released for commercial cultivation. Now, you know, the social media has a very important role in promoting technology. If you just Google Pusa Basmati 1718, you will find thousands of videos which have been produced by small entrepreneurs, farmers, and youngsters describing the features of these varieties. And that has led to fast spread of varieties. We have also developed a system of licensing varieties through a number of seed companies which are working in this area, and they have helped in fast spread of these varieties. In order to diversify the genetic base, we had collection from Northeast, particularly, and this is this seminar is focused on genetic resources. how we can do genetic enhancement or pre-breeding, where we can transfer the genes from a germplasm which cannot be readily used for commercial cultivation, but we transfer that into cultivable background, and then you incorporate that into high-yielding varieties. So a line called BAM72, which was resistant to bacterial leaf blight, it was crossed with 1121, and we followed the approach of QTL-seq. QTL-seq is a method by which you can identify the markers linked to the gene of interest. So following this QTL-seq approach, we identified the gene XA43, which is a recessive gene, and this gene, in this region where the gene was identified, 4 candidate genes have been identified, which we are now further trying to characterize. So this XA43T is new gene for resistance to bacterial leaf blight, which is recessive in nature, that comes from the land. Dr. Farmer director, in the farmers where this crop is affected badly by the neck blast disease, and neck blast is called gardan tod. Gardan tod means the farmers suffers a lot. The crop looks very good, but at the fag end of the crops, the neck blast infestation happens. Then the neck of the panicle will break, and these panicles will droop down, and the panicles will dry. So you will not have the grain filled properly. You will all have chaff. To solve this problem, we started a breeding program where we transferred several genes for blast resistance, and these are PI1, PI5, PETA, PI5, PI9, PI2, and PIB. And through molecular marker-assisted backcross breeding, we transferred these genes into Pusa Basmati 1, using Pusa Basmati 1 in all crosses as a recipient parent, and this is donor 1, donor 2, donor 3, donor 4, and donor 5. And now we have come out with the product which has got 1 gene, 2 gene, 3 gene, 4 gene, 5 gene, up to 7 gene pyramids in the background of Pusa Basmati 1. So these new lines of Pusa Basmati 1 would become a very important source for utilization in the breeding program for transferring these genes into other Basmati varieties. Here you can see the donor that we used in this case is IRBL9W, and this donor is so bad, the color of the pericarp is red. So this rice nobody will like. Its grains are chalky, on cooking it will swell. On contrary, the Basmati rice has got slim grains, it elongates on cooking, it has got good aroma, no chalky grains. So when we use this kind of donor, then always you have many undesirable characters, they come to recurrent parent, and that we have to eliminate through marker-assisted background selection. And following that procedure, now we have developed a number of lines which combine resistance to blast disease, the gene PI9. And one of such lines, here you can see Pusa Basmati 1, which is we have grown in Himachal Pradesh in Milan, which has a uniform blast nursery. All plants are killed. But here, all plants are killed. I would request the other participants to kindly mute their mic. So the PB1, when it is incorporated with PI9 gene, then nothing happens to these plants, where these plants are killed completely. So that's the power of single gene providing resistance. And one of such lines, Pusa Basmati 1637, has been released for commercial cultivation with yield of 28 to 35 quintals per acre. And our Director General ICR and Secretary there, Dr. Tilochan Mahapatra, are visiting the farmers' field. We followed the other disease, I say, is the other disease is the Bakane disease, which is caused by a fungus called Gibberella fusicori. And this fungus, after infection, produces gibberellic acid in the plants, and the plants will grow tall and ultimately they will dry. So you can see this is a field of 1121 variety which is infected with Bakane disease. All plants have grown tall and they are drying. Almost 25 to 30% infection happens in the field, looks very ugly. We have mapped a gene for resistance to Bakane, and now we are transferring this gene into 1121. The new lines will be available soon. So this is the one where we have transferred the gene QBK for Bakane resistance 1.2 in 1121 background. Also in 1509, we have transferred the same genes for resistance to Bakane because 1509, 1121, 1401, they are all highly susceptible to Bakane disease. So these are the new lines which have got resistance to Bakane, and at the same time grain and cooking quality similar to the original variety 1509 and 1121 and 1401. Another area that we have been working on is herbicide tolerance in rice, where we identified a mutant through EMS mutagenesis. And then we transferred that gene into Pusa Basmati 1509 and 1121. And after 18 days, now the herbicide imijathapir is sprayed here. And because of this spray, nothing happens to rice crop because it is resistant to imijathapir, but all weeds are killed. So you can see here weed killing starts happening. And the particular line which does not have this gene for resistance to herbicide, that rice plant is also killed. But wherever we have transferred gene, they are highly resistant. So now you can see the crop here. Rice is also killed and weeds are also killed. But whenever in rice we have got ALS resistant, herbicide tolerance gene, nothing happens to these plants. So now these lines are in advanced stage of testing. Here also you can see original 1509 is completely killed, but this one carrying resistance to ALS is fully surviving. But their quality is exactly identical to original variety 1121. And in addition to this, we are working on some short grain aromatic rices. Kala Namak is one of the short grain aromatic rices, which is a prized possession of Eastern Uttar Pradesh. It is protected under geographical indication like black rice of Manipur, which is also a GI protected commodity. But the traditional Kala Namak is very tall, up to my shoulder's height. And we started a breeding program to make it dwarf. And now the new generation's lines, you can see their height is up to my loin height, and this is just about 80 centimeters. This doesn't last, and it has got very high productivity. So we are trying to now push this particular material in Eastern UP. So I will not go into detail. Other aspects of basic research, we also worked earlier on golden rice through transgenic approach. But this line of work we have now discontinued. Since we are running late, I would like to close at this point of time, and I would like to thank all my colleagues, my students, my scientific colleagues, and also technical and supporting staff for their excellent support in the breeding program. Thank you very much. If there is any question for me, I will be more than happy to answer those questions.
Thank you, sir. Thank you, sir.
Thank you. Thank you.
The good lecture. Keynote lecture on crop. Actually, sir, about 5-6 years before, Labour Prime Minister asked to Asia scientists what is outcome of new technology. Then only single Basmati rice variety saved the Asia that is developed by the Dr. Ekasingji. So thanks to sir for giving this technology to Indian farmers. Thank you, sir. Then I request to Ekasingji sir to start next session.
No, continue with this session.
I would like to request Honorable Vice Chancellor to please permit me, because I have several other commitments. When I am free, my IRC is also going on, so I had to leave my Institute Research Council meeting and join this presentation. If you please permit me and somebody, maybe Dr. Shastri or somebody else.
Dr. Shastri. Thank you, Dr. Ekasingji sir, for your nice presentation.
Thank you, sir. Thank you.
And because actually you're a student, you're working. I'm very happy for the Chaghao, that is Black Prize, and because actually proofing is successful, and there's benefit farmers of the Northeastern region for the whole country. It is big achievements, I should say. And now actually, since you are very much preoccupied, have the time kindly. Otherwise, actually, I will handle for conducting this technical session. Thank you.
Thank you, sir. Thank you, Dr. Ekasingji, for acceding to our request. I know your time is very valuable. Well, we are very delighted that you gave your research findings and you shared. There are several questions coming, but then we will take up later on. Thank you, sir.
Thank you so much.
Very much.